Welcome back to the deep dive. Today, we are taking a dramatic shortcut right into the core of this cell. We're going beyond just, asking what genes are expressed. We're asking exactly where those RNA instructions are sitting inside the cell. It's a field called subcellular geography.
And it's so important because the physical location of an RNA molecule, it's not random at all. It really dictates its function and, ultimately the fate of the entire cell.
This feels like a huge leap forward. What's making it possible now?
It's really a technological moment. The old barriers are just crumbling. We're now working with these high-resolution spatial transcriptomics technologies, or XET. Think of things like MERFISH and SecFish plus ANA combined with really sophisticated sequencing methods.
Like sexscope or stereo-seq.
Exactly. And these platforms are giving us a spatial resolution down to 0.1, 0.2 micrometers. It's just incredible.
That's fine enough to see inside the different parts of a cell.
That's the precision you need, to actually measure gene expression within subcellular compartments.
And this isn't just some academic curiosity. This intracellular localization is vital. Can you give us a quick example of why this location is, well, everything?
Absolutely. So think about targeted delivery, right? Localized protein synthesis. The classic textbook example is beta-actin mRNA. This molecule, it isn't just floating around randomly. It's strategically placed right at the leading edges of fibroblasts.
The parts of the cell that are actually moving and extending.
Precisely. This makes sure that the structural protein, actin, gets built on demand right where the cell needs it to grow or move. If that mRNA is in the wrong place, the cell just loses its way.
And when that delivery system goes wrong, I imagine the consequences get pretty serious.
They can be catastrophic. We see it in several major diseases. Take Huntington's disease, for instance. A lot of researchers now believe that the neurodegeneration there is linked, at least in part, to disruptions in how mRNA is transported down the axon.
So being able to map these patterns is critical.
It is. Knowing if an mRNA is stuck in the nucleus, or maybe lost in the cytoplasm, or correctly positioned, that's key to understanding both basic biology and, where diseases come from.
Okay, so we have this explosion of amazing data, but the software, the computational tools, they haven't kept up.
Not at all.
Researchers were stuck with older frameworks, right? Things like Bento and SPRAWL.
Yeah, and they just hit a wall. You have to remember, those tools were designed for a much simpler time, for older imaging-based data.
Just points on a map?
Basically, yeah, point clouds. So when you try to feed them the really complex, high-volume data from something like sexscope, they often just fail.
Okay, let's unpack this. We are diving into ELA subcellular expression localization analysis, a new framework designed to solve this very problem. But first, what were the specific limitations of those older tools?
Well, there were a few big ones. First, just platform rigidity. As we said, they couldn't handle the new sequencing data. Second, and this is a big one, they had very low discovery potential, meaning they were limited to detecting only predefined patterns. So you had to tell the software what to look for if a gene had some weird, novel pattern.
The tool would just miss it.
It would completely miss it. So you're only confirming what you already suspect, not really exploring.
And I heard there were statistical problems, too.
Huge ones. Bento could only analyze one cell at a time, which is a big problem for statistical power. SPRAWL could look at multiple cells, but, well, simulation showed both of them had really, really low power.
How low are we talking?
We're talking a power of around 0.04.
Wow.
Which means they were failing to detect real, true patterns almost all of the time. We needed something much, much more reliable.
Here's where it gets really interesting. How LA overcomes this using statistical modeling.
The first thing LA does is really clever. It creates what's called a unified cellular coordinate system.
Okay.
See, cells come in all shapes and sizes, right? Oval, star, you name it. So you can't just use, say, absolute distance from the center.
It wouldn't be comparable.
It wouldn't be comparable. So ELA standardizes the geometry. It defines the center of the nucleus as a relative position of 0. And the outer boundary of the cell, that's always a relative position of 1.
Ah, so no matter if you have a giant liver cell or a tiny neuron, the nucleus is always 0 and the edge is always 1. It's like a standardized one-dimensional map of every cell.
Exactly. And that standardization is the key. Because now you can jointly model transcripts across thousands of different cells at the same time, no matter what they look like.
And that's where the massive boost in statistical power comes from.
That's it. It borrows information across all those cells.
But hold on. If you're mathematically sort of averaging all these cells together, isn't there a risk that you might miss a really rare but important pattern that only shows up in a few specialized cells?
That is a fantastic question. And that's where the statistical model itself comes in. ELA uses something called an over-dispersed, nonhomogeneous Poisson process. It's a mouthful, I know.
Let's break that down.
So the nonhomogeneous part is what lets the model spot non-uniform patterns. So areas where RNA is enriched or depleted along that 0 to 1 radius. It doesn't assume everything is evenly spread.
Okay, and what about all the noise? I mean, biology is messy.
It is, and that's what the over-dispersed part is for. RNA counts are naturally noisy. They vary a lot from cell to cell. This factor allows the model to account for that natural biological messiness, so it doesn't mistake noise for a real pattern. It controls the type-by error really well.
And you mentioned ELA can detect pretty much any pattern. How does it do that?
Instead of just looking for one or two things, it uses 22 different mathematical blueprints. They're called beta probability density kernel functions.
22 blueprints.
Yeah, think of it as one for every possible pattern you can imagine. Transcripts clustered right at the center. A perfect ring near the membrane, something in between. It just lets the data decide which blueprint fits best for each gene.
And that combination is what gives it such a high statistical power.
It is. That's why ELA achieved an average power of 0.68 in simulations compared to, the 0.04 from the older methods. It's just night and day.
So to recap for a second, ELA is this powerful, scalable tool. It works across new imaging and sequencing platforms, and it finds patterns where others fail, all thanks to this standardized coordinate system and some very smart statistical modeling.
That's a perfect summary. It gives us a reliable foundation to finally do some real discovery.
Okay, so let's move from the methods to the biology.
Oh.
ELA was used on 4 really different data sets: mouse liver, embryo, fibroblasts, brain tissue. That diversity means we can start looking for some general rules here.
Yeah.
What was the most consistent thing you found?
We found some really clear fundamental relationships. It seems like the location of an mRNA is intrinsically tied to two things: its size and its ultimate destination in the cell.
Okay, let's start with the ones that stay close to home, the nuclear-enriched genes.
Right. Key finding 1, nuclear enrichment. Genes that LA found clustered near the nucleus, either at the center or the edge, had one thing in common. They were consistently longer. Significantly longer gene lengths.
And that held up across different ways of measuring length.
It did. Average isoform length, total exon length, all of it. And what's more, these clusters were full of regulatory transcripts, specifically long non-coding RNAs, LNc-RNAs, and transcription factors.
So why are these long regulatory molecules hanging out near the nucleus? Is it just like a traffic jam of things waiting to be exported?
That's a big part of the hypothesis. It takes longer to transcribe and process longer genes. So the idea is that these long or regulatory transcripts are deliberately retained in the nucleus.
For what purpose?
It could serve a few. Maybe it acts as a buffer to reduce noise in gene expression, or maybe it's a reservoir that the cell can tap into quickly when it needs to.
Is there any direct evidence for that retention?
Yes. In this EXCO data, we found really strong support. These nuclear-enriched genes had a much higher unspliced to spliced ratio.
Which is the molecular signature of what?
Of an RNA that is still being processed in the nucleus. It's like the made in the factory sticker is still on. It hasn't fully left yet.
Okay, so the long regulatory stuff stays close to the source. What about the other end of the cell, out near the membrane?
That's key finding 2. Cytoplasmic and membrane enrichment. And this tells a story of localized production. The mRNAs you find out there frequently encode things like ribosomal proteins.
The building blocks for more ribosomes.
Exactly. Or, and this is really cool, they contain a specific code called a signal recognition peptide, or SRP.
Ah, I remember SRPs. They act like a zip code for the protein, right? To send it somewhere specific.
You got it. The SRP is a cellular zip code that directs the final protein into the secretory pathway to the ER, the Golgi, or the plasma membrane.
So how does that affect the location of the mRNA?
Well, the cell is incredibly efficient. If the final protein needs to end up at the membrane, the cell doesn't build it in the middle of the cytosol and then transport it. It moves the mRNA blueprint right next to the endoplasmic reticulum membrane.
So the factory is set up right next to the delivery exit.
Precisely. The translation happens right there, feeding the protein directly into the secretory pathway. It's just maximally efficient.
And the data back this up.
Overwhelmingly. In the liver data, the cytoplasmic enriched genes had an SRP-coded proportion of almost 30%.
And the nuclear ones?
Just over 10%. So it was nearly a threefold difference. It's really powerful evidence linking the physical location of the RNA to where its protein product needs to go.
And what's so crucial here is that these aren't just artifacts from one technology. These are robust findings across liver, brain, embryo, fibroblasts, all using different platforms.
Exactly. And ELA was powerful enough to see dynamic things too. In the fibroblast data, for example, it saw changes across the cell cycle. Genes in the G1 phase, when the cell is growing, were less likely to be stuck in the nucleus.
Compared to the other phases.
Yeah, compared to the S and G2M phases, when the cell is replicating its DNA, it suggests that the whole process of DNA replication somehow enhances this nuclear retention of certain transcripts.
So ELA is showing incredible versatility. It's working across Xenium, Merfish, StereoSec, all the big platforms. It really feels like a new standard.
It sets the baseline, for sure. But there is a limitation to keep in mind. LA currently focuses on patterns that are rotation invariant.
Meaning they only vary along that radius from the nucleus to the edge.
Right. Rings, clusters, things that are symmetrical. And that simplification is what helps boost the statistical power. But, biology is often highly asymmetric. As cells have a front and a back, there's polarity. So that raises a really important question for the future.
Which is?
Well, the localization that ELA reveals is only half the story. The location is tied to timing of mRNA metabolism, how it's exported, how it's degraded. The next big frontier here is integrating this kind of spatial analysis with functional measurements of mRNA metabolism.
Using technologies like SLAMSec, which can track newly made RNA.
Exactly.
So you'd get a full spatiotemporal map of the RNA's entire life, from its birth in the nucleus to its work location and then its eventual recycling.
Yes. By combining localization with the kinetics of metabolism, we'd move from a static map to a dynamic movie of the cell's entire regulatory architecture.
So we've seen how ELA has already uncovered these robust rules connecting gene length to nuclear retention and signal peptides to membrane localization. We'll leave you with this final thought. Once we can actually create that dynamic movie and track the full metabolism of RNA spatially, what other hidden cellular processes might finally be revealed by this unprecedented view into the cell's internal world?